Hi, everyone. I wanted to provide some information for session 4 the week of September 24th. So let's take a look at some of the tasks that are in that folder. Okay, so once again, the agenda is provided for all of the different tasks that you need to complete by the end of that week. So everything needs to be done by Saturday, September 29th. Okay, so all the links to all of the Edpuzzle videos are there. All of your Brain Pop videos are there with the username and the password. So you have to watch those videos, answer the questions, and email those results to me. What I want to do is take a look at the virtual lab that you guys are going to be working on. So there's a virtual photosynthesis lab, and there's a link here. And then there's a document here where you're going to collect some data and submit that. So let's take a look at the virtual lab. So to familiarize yourselves with some of the information that's on there. Okay, so if you click on that link, it'll take you to this site here. So there's a little animation here. And in this animation, you're basically taking a look at the effect of certain variables on the rate of oxygen being produced. So you want to see what variables are going to affect basically how much photosynthesis is happening. And the variables that you can manipulate on this animation are temperature. So you can change the temperature from 10 degrees to 25 to 40. So you might want to take a look at the effect of temperature on the amount of oxygen being produced. You can also take a look at the effect of light intensity. So you can slide this dial over here from 0 to 50 and see what effect do the different light intensities have on the amount of oxygen that's being produced. You can also change the color of the light. So similar to another investigation that you did a few weeks ago, where you can change it from white light to orange to green to blue. Okay, so you could take a look to see what effect the light color has. You can also take a look at the effect of dissolved carbon dioxide on the rate of oxygen being produced. So this is the standard setting here, and then if you click on the vial here, you can increase. So it's a little counterintuitive because when there's actually less of the water in here, it means that there's more carbon dioxide that the plant is exposed to. And then there's a little timer here where you can time and count how much photosynthesis is taking place in the form of oxygen bubbles. So if you see here that there are oxygen bubbles that are being produced there. Okay, so there's an accompanying worksheet with this where you would record some of the information. So you have this document here, okay, where you'll record all of that information. So let's say, for example, you wanted to take a look at the effect of temperature. So the question would be something along the lines of, what effect does temperature have on the rate of oxygen being produced? And then you would make a hypothesis on that. Which temperature do you think would be the optimal temperature that will produce the most amount of oxygen per minute? Okay, so you would make your prediction there. And then on the data table here, you would record some of the information here. So in this first column, if you're taking a look at the effect of temperature, over here you would type in temperature and then include the different settings, 10 degrees, 25, 40, if you're doing that. And then there's a table here to count the rate of oxygen being produced. So essentially what you would have to do is, so once again, like if you're taking a look at temperature, you can set it at the first temperature, 10 degrees, and then you can set the little timer here, and you can count how much bubbles are being produced per minute. I know it's a little tedious, so essentially you could count how much bubbles are produced in the first 10 seconds. Just multiply that by 6, in order to get how much bubbles are, are produced per minute. So you would do that 2 times for each setting and record that information here and find your average. Okay. So then you would also need to include what your independent and your dependent variable is. So once again, your independent variable is the variable that you, as the experimenter, is changing or manipulating. So in this example, it would be the temperature if that's the investigation that you want to do. But there are other variables that you can manipulate. You can manipulate light intensity if you want to take a look at that. You can manipulate light color. That could be your independent variable. You could manipulate the amount of dissolved carbon dioxide there. Okay. And the dependent variable for everyone is going to be the same. The dependent variable would be the amount of bubbles. The bubbles represent oxygen bubbles. So once again, this investigation is taking a look at photosynthesis and what variables affect how much oxygen is being produced. So your dependent variable would be the number of bubbles produced, oxygen bubbles produced per minute. And then your constants would be all of the other variables that need to be held the same. Because if you recall from some of the videos on scientific method, you should only change one variable at a time. Otherwise, you won't know which variable is having the effect in the experiment. So in the example, if we're taking a look at temperature, if that's the independent variable that we're examining, all of the other variables in the investigation need to be held constant. So for example, whatever the light intensity is for each setup, that needs to be the same. The light color needs to be the same. The carbon dioxide, all of those other variables would be held constant. Okay, and then after going through that virtual investigation, you would make your conclusion and see if your hypothesis was supported or not based on the data that you collected. Okay, so you'll complete this sheet and you'll upload it onto Blackboard. Okay, so that's the virtual investigation. Then there's some Edpuzzle videos. And then there's another document on here, a POGL exercise. So let me pull that up so you can see what that is. Okay, so POGLs, they stand for Process-Oriented Guided Inquiry Learning. And it's essentially a guided inquiry approach to help students develop an understanding of some concept. Okay, so ideally you would be working on this in groups. Potentially you can submit this as a group. I'm working on setting up some groups. If you want to do these tasks individually, that's fine. If you feel that you might need some support and would like to work in groups, we can set something like that. And because I do want to incorporate some group dynamic in the class to help facilitate some of the learning. Okay, so this is the first POGL exercise that you're going to be working on, and it's on photosynthesis and respiration. Okay, so the way POGLs are set up is that there's typically some diagram at the heart of the POGL And the diagram is designed in such a way to help students develop some sort of understanding of some concept. And in this particular POGL, is to help develop an understanding of photosynthesis and what's taking place, and cellular respiration and what's taking place. Okay, so take a look at the diagram here. Might look a little confusing if you're not sure what's going on here. But essentially, there are explicit questions. They're focused questions based on the diagram that are being asked. So I'll go through a couple just so that you can get a feel for it. Okay, so for example, the first question here, it says to refer to Model 1, and it says, in what cell organelle does photosynthesis occur? Okay, so you have to look at the diagram here. So up here is the section for photosynthesis, and it says that photosynthesis is occurring in this structure here of the cell. If you recall from some of the information from the other videos, this structure here is called the chloroplast. So photosynthesis is a process that occurs within the chloroplast of the cell. Okay, and then the second question is taking a look at, well, what are the three reactants that are needed for photosynthesis? Okay, so it gives this equation here that takes a look at the reactants that are needed. So you can disregard these coefficients here, but you need this, which is the formula for carbon dioxide. So that's one of the reactants. You need this H2O, which I'm sure everyone knows is water, and you need energy in the form of light. Okay, so you're going to go through these POGLs and respond to the questions. Okay, so for this particular POGL, there are 10 questions that all refer to this diagram here. Okay, so you're gonna respond to those and submit that onto Blackboard. And once again, I'm gonna work on setting up some groups if some people might not be familiar with with photosynthesis and respiration and how to interpret that. We can potentially set up some groups to get some additional assistance, and you can always feel free to email me. Okay, so that's the POGL exercise. There's also going to be a Kahoot on there at the end, and then lastly, you're going to do the discussion questions. Okay, so yeah, that's it for session 4. If you have any questions, concerns, issues, or anything like that, please feel free to send me an email. Okay, thank you.